안녕하세요, and welcome to the webinar. I'm Wing, and thank you for the kind introduction. And I'll be your field application scientist for GenScript's protein expression and antibody production portfolio. And for today's presentation, I'll be providing you with an overview of GenScript's proprietary and customized antibody generation for therapeutics, diagnostic, and basic research applications. Okay, we take pride in being the #1 gene synthesis provider in the world, and from the natural extension of our gene synthesis services expertise, we are well positioned to become custom antibody service provider and are honored to be voted and awarded as the most popular custom antibody supplier by SiteApp. We have extensive experience in custom polyclonal, monoclonal, anti-idiotype antibody, antibody modification, and antibody drug discovery. We have about 3,000 employees globally, and 30% of them holds a postgraduate level degree. GenScript have been cited in over 30,000 peer-reviewed journal articles, and we at GenScript offers a comprehensive service portfolio to scientists in more than 160 countries, ranging from gene synthesis services to peptide, protein, and antibody services, and a full array of COVID-19 solutions for vaccine development and beyond. So for today's presentation, I will give an introduction to the GenScript antibody platforms, some of the case studies that we have for the custom antibody cases, and we had in research, diagnostic, or therapeutic applications. And lastly, what are the other antibody-related specialized services that we have in GenScript? We are more than a professional service provider, and our highly skilled scientists may help shape your research program to the next level. Okay, first, let's start the presentation with GenScript antibody platforms introduction, many of which are state-of-art capabilities that are proprietary and unique to GenScript. So at GenScript, we are proud to be one-stop solution for all custom antibody needs. We capitalize on the animal's natural immune system to generate high-quality antibody leads. With high specificity and affinity, thanks to their intrinsic affinity maturation abilities for polyclonal and monoclonal antibodies. But how do we do that? GenScript has developed a series of proprietary antibody technologies to help ensure the success in generating high specificity and affinity antibody that can maximize your research potential. Our optimal antigen design program is using an advanced antigen design algorithm to optimize the likelihood of surface exposure and antigenicity. ImmunoPlus technology makes use of key immunomodulators to circumvent self-tolerance mechanisms and also allow B cells to generate high-affinity surrogate antibodies. Epitope binding is an independent antibody validation in which monoclonal antibodies are tested against one another to determine which antibodies compete for the same antigen epitope. GenScript also has a proprietary adjuvant system to quickly elicit a strong, durable immune response to generate high specific antibodies. Last but not least, the express immunization and high efficiency electrofusion allow antibody generation of speedy turnaround with large quantity and diversity. GenScript has established state-of-the-art animal facilities with ALAC International and OLOR certification. Our facility can provide a wide range of antibody services for individual and also bulk orders. The choice of polyclonal and monoclonal antibodies is highly dependent on the applications. Being able to recognize multiple epitopes with high tolerance for differences in antigen and are easier to produce, polyclonal antibodies are often more for novel targets or time-sensitive projects. In fact, we at GenScript have polyclonal packages that can allow delivery of antibodies in as fast as 45 days. Monoclonal antibodies, on the other hand, are the preferred alternative for well-established targets or when antibodies are required to recognize protein with specific post-translational modifications, such as phospho-specific antibodies, or when scientists require long-term reproducibility in high-stake projects such as in vitro diagnostic assays, vaccines, or drug development, etc. So monoclonal antibodies become a safer choice because there is always a new renewable and there is less batch-to-batch, and there is always batch-to-batch consistency, with less background and are highly specific. However, with all these advantages, it comes with a slightly higher price to produce, and also it sometimes may be too specific. That is, it is less likely to recognize a range of species. So to enhance the possibility for generating monoclonal antibodies successfully, and also to give the best value for monoclonal antibodies, GenScript uses time and tested technologies such as hybridoma technologies, and we also use new innovative technologies such as the B-cell cloning and also the single B-cell sorting, which enable us not to only accelerate B-cell selection but also capture a large B-cell repertoire. So to elaborate on the hybridoma technology we use, upon harnessing the animal's immune system to generate antibodies of high specificity and affinity due to their intrinsic affinity maturation abilities, we use reliable and time-tested hybridoma technology to immortalize antibody-producing B cells, producing hybridoma cell lines to generate high-quality monoclonal antibodies. Just by growing them, hybridoma cell lines provide an unlimited supply of antibodies. As a global leader in custom monoclonal antibody generation, we are constantly pushing ourselves to provide better quality and services to using advanced technology. And recently, GenScript has upgraded our monoRep technology and is now able to generate a larger number of high-quality rabbit monoclonals to maximize the success of finding monoclonal perfect for your specific research application, especially in difficult applications such as immunohistochemistry, therapeutics, in vivo and in vitro diagnostics, and also anti-idiotype antibodies. And due to the rabbit's superior immune system, they can generate antibodies with higher affinity, specificity, sensitivity, and stability than those generated in other animal species. Rabbits with robust and unique immune system also enable superior diversity antibody generation. That is, the rabbit is able to have a higher affinity, up to 10 to 100 times better compared to the rodents. In addition, we also have upgraded our upgraded monoRep technology, the rabbit monoclonal generation platform. We use the B-cell cloning approach instead of the hybridoma technology for antibody discovery. So this is a typical workflow of our upgraded monoRep, which involves the isolation, enrichment, and culturing of antigen-positive B cells from rabbit peripheral blood mononuclear cells, PBMCs, for screening and selection. Desired monoclonal clones are sequenced and recombinantly expressed, and through B-cell cloning, you can achieve rabbit monoclonals ideal for your research in research applications in as fast as 13 weeks. Many of these benefits of this approach include that we only get the PBMC, so the animals are not sacrificed. Not only so, it is fusion-free. It doesn't need to fuse with the with the myeloma cells, so it's fusion-free, and this can avoid the B-cell loss, and this gives us the ability to obtain a larger pool of up to 10,000 memory B cells. So, however, despite various advanced technologies, some researchers still require much more clones to prevent missed opportunities or a higher success rate, or they need it to be more time efficient in their antibody discovery. So our Beacon platform would be the answer. It is the solution for customers looking for high diversity antibodies and are willing to invest in high quality antibodies with good diversity, specificity, and affinity. Harnessing the latest optofluidics technology, Beacon combines the isolation and screening and evaluation of B cells on the chip with the integration of high throughput platforms for assay screening. GenScript has upgraded our lead generation capabilities by introducing a B cell sorting platform with a variety of in-chip screening assays available to identify positive antibody clones. In-chip assays include antigen-specific bead assay, as shown here, to recognize the antibody, or multiplex IgG capture assay to recognize the antibody, or reporter cell assay. So using the Beacon system from Berkeley Lights, GenScript could screen using multiple binding and functional assays to select only the most qualified antibody leads in 24 hours. And this shortens the screening process compared to around 12 to 14 weeks in the hybridoma technology, which is about 3 months, or within 2 to 3 weeks in the upgraded monoRep. The hybridoma technology can assess only less than 1% of the B cell repertoire, and B cell cloning and Beacon can both fully assess the antibody repertoire. This means that the antibodies discovered using these two platforms are likely to recognize a wider range of epitopes on the target antigen, increasing the chances of identifying rare antibodies for challenging targets that may fail with the hybridoma pathway. Having the B cell cloning and Beacon technologies enable us not only to accelerate antibody discovery, but also to capture a bigger B cell repertoire, thus better antibody diversity and having a higher clone number. So B cell cloning can lead to more sensitive clones when compared to the hybridoma technology. This is especially crucial for challenging projects. And we have data from across 9 challenging projects in GenScript, whereby we observed that B cell cloning yielded a higher positive rate and clone number than the hybridoma technology for the same project. So based on our wide base of customer feedback, B cell cloning have received the best feedback from our customers by having high clone number, diversity, and fast turnaround time, and high success rate with challenging projects, and yet, most importantly, at a cost-efficient price. With all these technologies and guarantees, GenScript has served a broad range of customers with good track record. We fulfill researchers' investigation on novel targets for drug and biomarker discovery, food and agriculture, industrial applications, and in vitro diagnostics. All these applications rely on high-quality antibodies to characterize, identify novel protein or enzyme targets. Our antibodies can detect the targets reliably with high specificity and affinity in a variety of research and development projects, including but not limited to immunofluorescence, Western blot, immunohistochemistry, ELISA, VAX, blocking assay, immunoprecipitation, positive, negative cell selection, and also in vivo functional assays. So here we would like to showcase some of the case studies that we have for our custom antibody generation platforms. So Colossoft and all in 2017 had used our PolyExpress services to generate a polyclonal antibodies for identifying a novel protein or claudin in the larval C. lamprey tissues. The monoclonal antibodies were used in Western blots here, and also immunocytochemistry for novel protein characterization. And using our antibodies, they managed to define its localization and its abundance in larval C. lamprey tissues. Okay, next, we show another research application in an article published in the Science Journal by Han and al. Tylenol had used our customized polyclonal and monoclonal antibodies products and services in immunocytochemical study and also Western blot to demonstrate the localization and the role of SHISA7 in controlling the GABA receptors abundance in the synapse in the central nervous system. The antibodies were very specific with low background as shown here. And this enables the identification of SHISA7 in hard-to-find locales such as the postsynaptic densities. And this research has significant physiological and clinical relevancy for the central nervous system mechanism of action understanding. This Nature Medicine paper was published by the Vaccine Research Center in collaboration with GenScript. The research was trying to develop a HIV vaccine via the HIV-1 fusion peptide and investigating the potential of using neutralizing antibody against this fusion peptide as a therapeutic strategy. One monoclonal antibody generated as part of this project was VFP-1.01FP, and this was further characterized in crystallography experiments to interrogate the structure and function of this antibody against the VFP-1.01FP. Alanine scanning to determine the residues necessary for the antibody function, and we also use surface plasmon resonance to determine the antibody binding strength. Outcomes of this project were we were able to look at and decipher the antigen prototype can be a good vaccine technology. Secondly, the neutralizing antibody can develop into a therapeutic candidate in HIV disease. Here again another research application showing our rodent monoclonal antibody product to detect survivin as a biomarker for tumor. And we enable this monoclonal anti-survivin antibody to develop into a therapeutic candidate for tumor control, as shown here by a decrease in the growth size of the tumor upon injection of the monoclonal antibody, and also an improved survival outcome upon the injection of this monoclonal antibody. In this study conducted by GenScript, mono-Rab technology was used to develop an anti-small compound antibody using ractopamine coupled to human serum albumin as an immunogen. Antibody developed here is specific to ractopamine while showing little cross-reactivity with similar compounds including salbutamol and also clenbuterol. Here, I would like to introduce a great example of GenScript commitment in joining scientists in the fight against COVID-19. GenScript leveraged the unique advantages of hybridoma technology, the mono-Rab B-cell cloning technology, and the Beacon platform to generate high affinity, high diversity antibody against antigens, including the spike protein receptor binding domain as shown here. The spike protein has been recognized as a popular vaccine target to develop COVID-19 vaccine. Many therapeutics are also leveraging the functional neutralizing antibody that can block the RBD-ACE2 interaction, thus reducing the virus infectivity. So how do we go about the process in generating the spike RBD rabbit monoclonal antibodies using the Beacon system? So after rabbit immunization, we generate a single cell suspension from one rabbit spleen and subsequently concentrated the antibody secreting cells using antigen and IgG positive B cell enrichment process. And altogether, we imported over 15,000 cells onto the Beacon system and subjected these cells to antigen positive screening using the antigen beads conjugated with RBD. Eventually, we identified a total of 142 RBD positive hits. In the parallel comparison experiment, single B cell suspension was also subjected to hybridoma process to compare head-to-head for hybridoma and Beacon platform. So not only monorac beacon method was able to introduce high affinity antibodies, we also achieved high diversity. This is a second experiment to compare the rabbit beacon versus the hybridoma platforms. And as you can see, a total of 8 monoclonal antibodies were obtained using the beacon experiments compared to 3 from the hybridoma approach. The 8 monoclonal antibodies from the beacon platform also had a higher diversity that spanned across 4 epitope beams, while all hybridoma antibody was in the same epitope beam. The high antibody diversity could successfully enable downstream applications, making them excellent candidates for IVD and other assay development or potential further development into therapeutics, thanks to the many candidates having a neutralization efficacy. And how do we know that? We evaluated the neutralization ability by evaluating the reduction of the luciferase expression within the ACE2 stable cells. Listed here in the bottom right panel, you can see our top 4 rabbit monoclonal clones with superb neutralizing potency. Many of the antibody leads are very well received by the research and industry communities for various applications like assay development and also therapeutic antibody licensing. As not only so, a higher diversity was also observed even compared to the beacon technology. A total of 16 antibodies were obtained with the B-cell cloning approach compared to the 8 monoclonal in the beacon and 3 from the hybridoma. And the 16 monoclonal antibodies from the B-cell cloning also had the highest diversity to span across 11 epitope beams. The high antibody diversity could successfully enable multiple downstream applications. As many of you may be interested in therapeutic antibodies generation, here we show our track record in delivering high-quality anti-idiotype antibody for pharmacokinetic studies and also immunogenicity assay use. Anti-ID antibodies are powerful tools in the pharmacokinetic studies to measure the antibody drug level in the patient sample. The anti-ID bridging format highlighted in the green box is extremely popular for high specific antibody drug detection, especially this antibody drug can always be at a rather low concentration. Anti-ID antibodies can also serve as a positive control or reference standard in immunogenicity assays to identify anti-drug antibody characteristics that can potentially explain the unintended side effects of an antibody drug. But given the important roles of anti-ID antibodies, scientists would like to generate this, but oftentimes scientists encounter various challenges during the anti-ID antibody production. Firstly, the CDR region of an antibody drug may not be very immunogenic, thus very little amount of the antigen-specific anti-ID antibody can be developed. Secondly, even there might be antibodies elicited, majority of the antibody may be FC region targeted, so less hits will be at the CDR. So scientists must always spend time and resources to optimize the clone selection from numerous anti-ID antibodies. As mentioned, anti-ID bridging ELISA is extremely popular for high specific antibody drug concentration detection in patient samples. So researchers love the high specificity as an advantage of using the anti-ID bridging ELISA format as shown here. However, oftentimes it is very challenging to obtain successful anti-ID pairs as in here and here. But thanks to many years of expertise, GenScript has developed the domain expertise in developing anti-ID pairs. Sandwich ELISA was performed to select an anti-ID pairs that could largely increase the success rate of immunoassay development. As shown in the right panel, monoclonal antibody 2 is able to pair with 4 other clones. Here 1, 3, 4, 5 for anti-ID bridging ELISA development. GenScript also have a good track record in developing anti-ID monoclonal antibodies against CAR-T. The CAR SC-FE region of the CAR is sequenced to generate a recombinant SC-FE. The recombinant SC-FE is then used as an immunogen in GenScript monoRep technology services to generate a mature anti-ID monoclonal antibodies against the original CAR. And we are proud to have the recognized track record to achieve high-quality antibody against CAR-T, and also end-to-end one-stop service for antibody generation to immunoassay development. Last, I would like to add a few antibody-related specialized services to further fuel your downstream development needs. As we just mentioned, GenScript is proud to enable end-to-end solution for antibody generation to immunoassay development for a broad range of antibody projects. We provide custom ELISA kit development for standard kits such as sandwich ELISA, direct-indirect ELISA, competitive ELISA, and also specialized kits such as pharmacokinetic ELISA, anti-drug antibody ELISA, and also whole cell protein ELISA. In addition, GenScript also offer antibody conjugation and labeling with biotin, HRP, and there are also many other fluorophore options. We offer many additional services such as antibody purification and also can scale up antibody production that I will cover in the coming slides. After you identify an antibody lead, one can improve its physical or functional characteristics and also properties, and thanks to the synthetic biology, scientists can engineer a library of different mutations and screen them to isolate the best antibody candidates. GenScript has established a pipeline for antibody engineering to further optimize your antibody lead quality. That is to improve specificity, stability, solubility, and affinity, or to remove hotspot degradation. The protein engineering also uses the same principle to optimize enzyme functions and thermal stability, and also allow better folding for new formatted fusion proteins. The antibody engineering capability can further improve our antibody lead quality, not only relying on animals' natural maturation, but also provide a systematic approach to holistically evaluate opportunities to optimize antibody function. Precise Mutant Library, PML, is a powerful tool to make antibody engineering possible. In this study, the objective is to establish a saturation mutagenesis library for 63 sites to all 20 amino acids. So the next generation sequencing results show that the ratio of amino acids at each site here at panel 2. We observe equal distribution of amino acids across all the sites, and there is no stop codon introduced. Precision control because this can minimize downstream screening burden. Imagine we have a 63 to the power of 20, that is about 1,200 candidates to screen, and this is no easy job. But here at GenScript, we introduce our proprietary platform, which is the fast screening of expression and biophysical properties and affinity, FASIBA. It is a throughput technology to enable us to screen for antibodies with the best antigen binding affinity, high expression level, and also thermal stable. As you can see, the top 5 candidate antibodies shown here have superior binding as shown by the KD and via surface plasma resonance SPR compared to the wild type antibody. So now that you have a good antibody candidate, you may want to express it in large quantity for cellular or in vivo evaluation. Our high-density expression system allows us to increase the yield of transient mammalian expression by 2 to 6-fold using GenScript's proprietary cocktail solution. These cocktails can boost the cell growth and regulate cell cycle, and thus achieve higher cell viability, which is essential for high antibody yield. Here we showcase a gram level recombinant protein, recombinant antibody expression using this transient expression system, high density HEK293F. On the left, it is the SDH, SPage, and the Western blot for the QC results. And on the right panel C, is the size exclusion chromatography, which shows that we were able to obtain 99% purity for this antibody. So with this, with all the information put together, I hope that our end-to-end antibody pipeline can best fit your research needs and will help you achieve your research goal without going through all the hurdles one can encounter during antibody discovery for therapeutics, diagnostics, and basic research applications. And with this, I thank you. 감사합니다.